This is the third video of the cell death pathways lecture. In this video, we will talk about two different types of neuronal cell death pathways, necroptosis and parthanatos. So, necroptosis is it's a little complex cell death pathway because it's kind of like a combination of necrosis, but it's a program necrosis pathway that includes different activated and cascades that makes it very different from necrosis. So necroptosis can be considered a type of cell program necrosis. Um, the very the importance of necroptosis resides in that it is induced by death receptors, mimicking what we've seen for extrinsic apoptosis. One of the most important receptors that participate in necroptosis is the tumor necrosis factor receptor. This pathway requires kinase activity of the receptor interacting protein RIP1 and RIP3, and it's considered caspase independent, but that is not appropriate. So it actually requires caspases to be inactivated, which is a different thing from being independent. And its execution involves the active disintegration of mitochondrial, lysosomal, and plasma membranes. And it's highly associated to inflammatory diseases. So, necroptosis participates in the pathogenesis of diseases, including ischemic injury, neurodegeneration, and also viral infection. And here we have a diagram of the tumor suppressor or tumor necrosis factor receptor signaling pathways. The first scheme is what we've seen for the activation of the extrinsic apoptotic pathway, in which we have a ligand that receptor that is linked in the intracellular space with a pro-caspase, and upon binding of the death ligand, then caspase 8 get activated. So this is slightly more complex than what is depicted in that diagram, and that complexity is reflected here, in which this FADD plus caspase 8 are also associated with other receptors, RIP1 and RIP3 and TRAD. So in the case of apoptosis, for instance, when caspase 8 is activated and gets cleaved, that activation leads to inactivation of RIP1 and RIP3. And that is what differentiates the pathway into apoptosis. In the case of necroptosis, caspase 8 is not activated. So the complex is the same, but caspase 8 is not activated, as we can see here. As a result, these kinases are active and they can promote phosphorylation of different substrates that end up in promoting the necroptosis pathway. So we're not going to talk more about necrosis program and necroptosis, but just so you know, there is way more complexity in every cell death pathway, way more than I can show you in this video. But yeah, the biggest difference between necrosis and necroptosis is that in the case of necroptosis, this is ligand dependent, and there are certain specific enzymes that are activated as part of this programmed cell death pathway. So this is a different cell death pathway called Parthanatos, and it is relevant, or it's activated upon specific types of DNA damage. So Parthanatos or PARP1 dependent cell death, which is the poly-ADP ribose polymerase 1, and this is a DNA repair gene, does not require the mediation of caspases for its execution, as we've seen with intrinsic apoptosis that is activated in the presence of DNA damage. But this is clearly mechanistically dependent on the nuclear translocation of the mitochondrial associated apoptosis inducing factor AIF. So what this happens is that when PARP1 is activated upon DNA damage, it causes an accumulation of poly-ADP ribose that is released into the cytosol, and that causes the mitochondrial AIF released into the cytosol. This protein has the ability to recruit the macrophage migration inhibitory factor, which is a protein that has nuclease activity. And this complex translocates into the nucleus, binds DNA, and the nuclease activity of the MIF protein degrades and fragmentates the DNA, causing cell death. There are different ways of inhibiting this process. In this paper that is referenced here, the authors tried different opportunities, such as generating a MIF binding mutant or knockdown of MIF, in which there is no interaction with the AIF protein. So even if there is DNA damage and PARP activation, AIF cannot translocate into the nucleus, and that results in cell survival. And there are other types of mutants they have created, such as MIF nuclease deficient mutant, in which the complex can be translocated into the nucleus, but the MIF protein can no longer degrade the DNA or fragment or cut the DNA. So this is actually a process that can be stopped by developing strategies or therapeutic strategies that can target MIF and inhibit it or prevent its binding to the AIF protein. So in this summary slide of the different cell death pathways, I wanted to emphasize the differences between the 4 different cell death pathways that we've seen in the last videos. And I kind of like classify them based on 5 different characteristics. One is chromatin condensation, nuclear fragmentation, apoptotic bodies, caspase activation, and whether or not they can be considered programmed death. So for necrosis, there's like all of these factors do not happen in this situation. And you can see sometimes, as I mentioned earlier, necrosis being classified as a non-programmed cell death pathways. However, excitotoxicity caused by glutamate, it's a way of inducing necrosis in a programmed manner. So that's why I added here that sometimes can be considered programmed death. Then for apoptosis, all these characteristics are do happen in apoptosis. And then there's kind of like a gray area between necroptosis, parthanatos, and necrosis. So this will be the biggest differences between these 4 types. And then there are specific differences that we've covered in each of these slides in the previous videos. And finally, I leave you here with a short quiz to revise the most important concepts about this video. We will see this in class, and we will talk about any other specific or important concepts that have been revised in this video.